Bismillahirrahmanirrahim. Today we are going to learn about the sense of taste or gustation. The sense of taste or gustation is a special sense that is pursued by tongue. To know about the functional anatomy of tongue and the mechanism of gustation, we are going to the first slide. In this slide, we can see the structure of tongue. This is the apex of the tongue. This is the body of the tongue, and this is the base of the tongue. The dorsal surface of the tongue shows large number of pimple-like projection you can see over here, and these pimple-like projections are called papillas. These pimple-like projection or papillas are having four types. I'm going to write the four types. Number one, the circumvallate papilla. Number two, fungiform papilla. Number three, foliate papilla. And number four, the filiform papilla. Four types of papillas are there. The green color papillas over here at the base of the tongue, which are arranged in V shape, are the circumvallate papilla. These are the circumvallate papilla, which are the average number of circumvallate papilla are 12. The fungiform papilla is represented by red color, which are present throughout the body of the tongue. That is the fungiform papilla. The foliate papilla is present on the lateral side of the middle portion of the tongue. These are the foliate papillas. These are the foliate papillas. Now going to the detail. In this diagram, we can see various types of papillas. Now going to compare the papillas which are present on the surface of the tongue. This is the circumvallate papilla, which is represented by green color, which is arranged in V shape on the base of the tongue. This circumvallate papilla is represented in magnified form over here. The average number of circumvallate papilla is 12. This is the fungiform papilla. The fungiform papilla is having the shape of the fungi, a shape of mushroom. This is the mushroom shaped. That is why this is called fungiform papilla, and this is present throughout the surface of the body, which is represented by red color over here throughout the dorsal surface of the tongue. This is the foliate papilla, which is having the shape of leaf, and this is present on the lateral side of the middle portion of the tongue. This is the foliate papilla. And this is the filiform papilla. Filiform papilla is having the shape of a thread or hair. This is thread-like or hair-like, and this is present all over the surface of the tongue. This is not responsible for detection of taste. I am going to tell you later. The circumvallate papilla is called circumvallate papilla because this is rounded in shape and surrounded by a wall. Circumvallate, the circular shape surrounded by a wall. That is why this is called circumvallate papilla. The circumvallate papilla contain large number of taste buds. The average number of taste buds in circumvallate papilla is 100. The fungiform papilla also contain large number of taste buds. The average number of taste buds in the fungiform papilla is 10 to 20. The foliate papilla contain from 5 to 10 taste buds, but in most cases, in majority of cases, when an individual reaches the adulthood, he gets rid of all the taste buds which are present inside the inside these the foliate papilla. So the foliate papilla do not contain any taste bud after the adulthood. Now coming toward the filiform papilla is responsible for the grasping of food, for rolling of food. This is responsible for injuring other animals also, and this is also present in very strong position in the tongue of some carnivores. This is not having any taste bud, and um, this is not having any role in the detection of taste because this is not having any taste bud. Now coming toward the next slide. Now going to compare or magnify the fungiform papilla. The fungiform papilla is magnified over here. This fungiform papilla contain average number of 10 taste buds. You can see over here 10 taste buds are present, and the shape of the taste bud is just like the bulb. So the fungiform papilla contain the taste bud throughout its surface, but the circumvallate papilla and this papilla contain the taste bud on the lower side of the papillas. Over here, large number of taste buds are present. Over here, large number of taste buds are present. But over here and over here, no taste bud is present. Similarly, over here on the upper surface of the papilla, there is no taste bud. Taste buds are present on lower side, while on the fungiform papilla, throughout the surface of the fungiform papilla, taste buds are present. The taste buds are present. Each taste bud is having a pore that is called taste pore, and taste pore lead to the cells of the taste bud. I'm going to show you in detail about the taste bud. This is a taste bud. You can see that this taste bud contain large number of cells. Large number of cells are present in the taste buds. The shape of the taste bud is just like the bulb. This is having bulb shape. All around the taste bud, the epithelial cells are present. Over here, large number of epithelial cells are present. This is the taste pores, and the taste pores are exposed to the taste hair. These are called taste hair. So inside this bulb, you can see large number of cells. The four types of cells are present inside this bulb. I'm going to show you. The type 1 cell, this is the blue color represent the type 1 cell. The type 1 cell is present on the lateral side of the bulb, the type 1 cell. This is the type 1 cell which is not having any hair, that is not having any cilia or hair or taste hair. This is the type 1 cell which is having hair. This is responsible for the detection of taste. This is the type 2 cell which is also responsible for the detection of taste, and this is the type 3 cell which is represented by red color. This is also responsible for the detection of taste. The type 1 cell do not detect any taste because this is not having any receptor for the detection of taste. This work as a glial cell, just like neuroglia, just like the supporting cell, just like the Schwann cell, just like the oligodendrocytes of the which are present all around the neurons. So this work as a glial cell. This is the supporting cell. The type 1 cell is responsible for the detection of salty taste. The type 2 cell is responsible for the detection of sweet, bitter, and umami taste. The type 3 the type 3 cell is responsible for the detection of sour taste. Similarly, the type 4 cell working as the progenitor of the cell. These are called basal cell because all the cells in the taste buds are originated from the type 4 cell. That is why this is also called as progenitor cell or the basal cell, because all the cells are based on these blue color cell, basal cell. Similarly, coming to the this special type of cell, that is SHH plus cell. This is also called the Hitchcock signaling cells. The Hitchcock signaling cell abbreviated as SHH plus cells. This cell is originated from the basal cell. This is also originated from the basal cell, and this cell is the precursor of all the taste cells, excluding the type 1 cell. Excluding the type 1 cell, all the taste cells are originated from the Hitchcock signaling cell. This is called Hitchcock signaling cells because this cell contain large number of hedgehog signaling protein, and hedgehog signaling protein are only and only present in those cells which undergoes differentiation. So the hedgehog signaling is only the speciality of differentiable cell. All those cells which are going to be differentiated contain the hedgehog signaling protein. So due to the presence of hedgehog signaling protein, these cells are called the hedgehog signaling cells, and these are the precursors of type 1, type 2, and type 3 cells. Now, coming towards the next slide. In this slide, we can see that each of the test cell is supplied by the dendrite of the special type of neuron. This is gustatory neuron. This gustatory neuron gave the dendrite or provide the dendrite to these test cells and get the signal from these test cell. How it get the signal? Actually, the test hair are exposed to the taste spore, and taste spore are exposed to the food items. So so all the food which is in dissolved form goes through this taste pore. When the food item or any entity come in contact with the taste hair, the taste hair contains special type of receptor which detect a special type of taste. So the as I have told you that type 1 cell is responsible for the detection of salty taste. Similarly, the type 2 cell is responsible for three types of taste, and one type of taste is detected by the type 3 cells. So all the food which contain different flavor or different taste can be detected by these cells. How these are detected by these cells, I'm going to show you in detail. First, I would like to write these types of cells, and to make it simple, uh, the type 1 cell, the type 2 cell, the type 3 cell, the type 4 cell. The type 1 cell having cilia, having hair cell. The type 1 cell which is having the taste hair, that is responsible for the detection of salty taste. The type 2 cell which is also having hair like projection, that is responsible for the detection of sweet, bitter, and umami taste. The type 3 cell that is responsible for the detection of which taste? That is responsible for the detection of sour taste. And the type 4 cell is not responsible for the detection of taste. This is basal cell, and this work as the progenitor of cell. Progenitor of cell, because all the cells are originated from this type 4 cell. So the type 4 cells, I'm going to show you. The umami taste is a special type of taste that is also called the savory taste. The umami taste is also called what? This is also called savory taste. And savory taste is a special type of taste. The umami is the Japanese word that means the delicious. And this is the taste of the monosodium glutamate. And in majority of cases in Asian foods, the umami taste is there because of the presence of China salt. China salt is having the umami taste. Now coming to the detail. Now going to show you the detail of the taste cells. Each of the taste cells is having the hair-like projection that is called taste hair. This is the magnified form of the taste hair. This is the magnified form of the taste hair. Each taste hair contain large number of large number of receptor, but special type of receptor are present on the separate or respective taste cell, because the for example the type 1 cell contain or the hair of the type 1 cell contain the ENAC receptor. This is called epithelial sodium channel receptor, or this is simply called epithelial sodium channel. The epithelial sodium channel are only present in the type 1 cell having the hair, and the hair of the type 1 cell. So what is the function of epithelial sodium channel? The epithelial sodium channel is responsible for the influx of sodium. When the food containing sodium, or when the food containing salt come in contact with this cilia, it come in contact with this channel also. So this channel allow the influx of that sodium from external environment to the internal environment. When the sodium get enter from external environment to the internal environment, the positivity of the taste hair or the positivity of the cell increases, which leads to the generation of electric potential, which leads to the generation of action potential, and that action potential is transferred towards the base of the cell, and that secrete neurotransmitter toward the dendrites of the neuron, that generates signal, or that transfer the impulse to the cell body of this neuron, of gustatory neuron. So the epithelial sodium channel is responsible for the detection of salty taste, and this is only present in the type 1 cell. Similarly, the type 2 cell contain the G protein coupled receptor, this type, the G protein coupled receptor, this type, and this type of receptor. These three receptors are present in the type 2 cell. I'm going to show you in detail. This G protein coupled receptor is the G protein coupled receptor having CAMP as the second messenger. All of the G protein coupled receptor are having second messenger. So the second messenger of this G protein coupled receptor is the CAMP, and adenylyl cyclase is the enzyme which lead to the generation of this CAMP. So this G protein coupled receptor is responsible for binding with the sucrose or glucose. This bind with sweet items, with the entities, all those entities which are having sweet taste. So when it bind with the sucrose, it undergoes the conformational changes, and this conformational changes lead to the internal effect, which lead to the generation of CAMP, and that CAMP is responsible for the generation of action potential, which is transferred toward the gustatory neuron. So, and this is responsible for the generation of this is responsible for detection of sweet taste, and this G protein coupled receptor with the CAMP as second messenger is present in the type 2 cell, that is responsible for the detection of sweet taste. This G protein coupled receptor is the G protein coupled receptor having IP3, or inositol triphosphate, is second messenger. And this second messenger is cyclic adenosine monophosphate. This second messenger is inositol triphosphate, and this inositol triphosphate is responsible for generation of action potential. So this G protein coupled receptor is responsible for binding with the bitter item, all those compounds which are having bitter taste. For example, the quinine is the standard one. So when it bind with the bitter item, or any bitter taste element, it undergoes the conformational changes, which leads to generation of IP3 inside this cell, and that IP3 is responsible for generation of action potential. Similarly, this metabotropic glutamate receptor is responsible for the detection of umami taste. Actually, this metabotropic glutamate receptor is responsible for binding with monosodium glutamate. Monosodium glutamate is the formula of cyanide salt. When it combined with the China salt, or when it combined with the monosodium glutamate, it undergoes conformational changes, and similarly is the case with the metabotropic glutamate receptor. The exact mechanism of this is unknown, but still this leads to the generation of various types of second messenger, which leads to the increase in calcium concentration inside the cell, which leads to generation of action potential inside the cell. So these three types of receptors are present on the type 2 cell, which is represented in the green color. Now coming towards the type 3 cell receptor, that is the proton channel. This proton channel is present in the type 3 cell, and type 3 cells are represented by red color over here. The cilia, or the taste hair of type 3 cell, contain large number of proton channel, and proton channels are responsible for the detection of sour taste. Actually, all the acidic food or all the acidic item, all the acids have the ability to dissociate into hydrogen ion, which give hydrogen ion and dissociation. When the hydrogen ion come in contact with this channel, this the influx of hydrogen ion take place, and influx of hydrogen ion only takes place with the help of this proton channel. When it goes inside the cell, when it goes inside this test here, what happens, the positivity of the cell increases, and when the positivity of the cell increases, it leads to generation of action potential. So the proton channel is present in the proton channel is present in the type 3 cell, and that is responsible for the detection of soul taste. Now I'm going to tell you the threshold of these taste receptor. The threshold for the sweet taste and for the salty taste is very low. Threshold for the taste mean that how much concentration, below how much concentration these receptor will not be able to get the action potential, or to make the action potential, or to be stimulated. Number one, the sweet taste, the salty, the umami, and the not umami, the sour, and the bitter. The umami is unknown still now. So the threshold for the sweet taste, the standard for the sweet taste is used as sucrose. Sucrose is used as standard for sweet taste. Similarly, for salt, the NaCl is used as standard. Similarly, for sour, the HCl is used as standard. Similarly, for bitter taste, the quinine is used as standard. Quinine is an alkaloid. So the threshold for the sweet taste is considered as 0.01 molar. The threshold for the salty taste is also considered as 0.01 molar. The threshold for the sensation of taste for the sour taste is considered as 0.0009 molar. Similarly, for quinine or for the bitter taste, this is considered as 0. Penta 0.0000009 molar. So it means that below 0.01 molar concentration of any sweet element, this receptor will not be able to detect that taste. If the concentration of the solution having sweet taste is lower than zero point 0.1 molar, then this receptor will not be able to detect that taste. And similar is the case with the sodium. If the concentration of sodium is lower than 0.01 molar, then the salty taste will not be detected by the epithelial sodium channel. Similarly, the sour taste, for the sour taste, this is a bit low because this can be written as 0.001 by rounding up. So for the sour taste, this is also very low. For the bitter taste, this is extremely low. In extremely low concentration, the bitter taste can be detected 0.009 molar. So this is very low. This is what is the logic behind this low concentration, because you know that large number of sweet item and large number of salty item are not hazardous for our health, are not dangerous or harmful for our health. And you know that majority of the bitter and sour items are not good for our health. Majority of the medicine, majority of the drugs which we use, which we consume for which we take for to avoid various health anomalies, are bitter or sour in taste. For example, the quinine, a large number of medicine which is bitter in taste, so that can be detected in very small concentration. Why? This is a protective phenomena, because if the food contains some poison, if a food contains some alkaloids, or food contains some element which is bitter in taste, which is not good for our health, that is not nutritional. You should know about this, that the bitter element are non-nutritional. Large number of bitter element, majority of the bitter element are non-nutritional, and majority of the sweet element or salty element are nutritional. So to avoid the consumption of non-nutritional agents, the concentration of this or the threshold of the bitter taste is very low. This is a protective phenomena. This is what we call the beauty of God, that the threshold for the bitter taste is very low. Now coming towards the next slide. In this slide, we can see that when the action potential is generated by the taste cell that is transferred towards the dendrites of the gustatory neuron in the form of various neurotransmitter. The majority of in majority of cases, the neurotransmitter secreted by these taste cells are the serotonin. The serotonin are working as neurotransmitter which activate the dendrite, which make the action potential. Then this the dendrite of the gustatory neuron, and this gustatory neuron goes in three groups. Three types of gustatory neurons are there, which are three types of nerve are responsible for the detection of taste. I'm going to tell you in the next slide. In this slide, you can see that the tongue contained three groups. This is one group of neuron, this is another group of neuron, and this is another group of neuron. The nerve fiber which get the signal, which get the taste signal from the body and from the apex of the tongue, from the papillae of the body and the apex of the tongue, which get the signal from the taste buds from the apex and the body of the tongue. That is the nerve number 7, and that is called facial nerve. This goes towards brain, and this get the signal or get the taste signal from this area of the tongue. Similarly, this the nerve number 9, that is also called glossopharyngeal nerve. This is very important nerve, glossopharyngeal nerve, that get the signal from the base of the tongue and also from the taste buds which are present in the pharynx and the upper part of the pharynx of the behind the tongue. So it gets the taste signal from the taste buds which are present on the base of the tongue and from the pharynx of the body. So the third group of nerve, that is called vagal nerve, that gets signal from the lower part of the pharynx and from the esophagus also and from epiglottis also, because the upper part of esophagus also contain taste buds and the lower part of the pharynx also contain the taste buds. So from the pharynx and the esophagus, the taste signal are pursued by the taste signal are transferred by the vagal nerve toward the brain. Now going toward the brain. So the first order neurons from the tongue goes in three groups towards the medulla oblongata. Inside the medulla oblongata, a nucleus is there for the reception of first order neuron. This nucleus is called gustatory nucleus. The gustatory nucleus is further, this is present inside a big nucleus that is called tractus solitarius. But from this gustatory nucleus, the second order neuron arises, which goes toward the thalamus. Inside the thalamus, a special nucleus is there that is called posteroventral nucleus, which work as relay station for the second order neuron. So the second order neuron goes to the posteroventral nucleus of the thalamus. From the posteroventral nucleus of the thalamus, the third order neuron arises, and that goes toward the taste center. Now going toward the detail of the taste center. This is the taste center or gustatory center. This is present inside the cerebral cortex. In which area of the cerebral cortex I am going to show you. This is the frontal lobe of cerebral cortex. This is the parietal lobe of cerebral cortex. This is occipital lobe, and this is the temporal lobe. This is the central sulcus. Behind the central sulcus, there is an area, there is a gyrus that is called post-central gyrus. Post-central gyrus, I'm going to show you. This is the post-central gyrus, and this post-central gyrus is responsible for the reception of the somatosensory sensations. So below the post-central gyrus, below this somatosensory area over here, there is a lateral sulcus. This lateral sulcus going in between the lateral sulcus and the parietal lobe over here below the somatosensory area, the taste center is there. I'm going to show you in detail. This is the lateral sulcus, and this is the parietal lobe, and this is the temporal lobe. This is the operculum. This goes inside. This is also part of cerebral cortex. This is the operculum and this is the insula. So the taste center is present partially in the insula and partially in the operculum. Where in operculum, this is the taste center or gustatory center. This is present partially in the insula of the cerebral cortex and partially in the operculum of the cerebral cortex. Where in the insula or where in the you should be quite clear that this area of the cerebral cortex is the somatosensory area, and very near to the insula, very near to the operculum over here, this area is responsible for the detection of somatosensory sensation from the tongue. So this is for general tactile sensation, and this area is gustatory center, which is also getting the action potential are getting the taste sensation from the tongue. So this taste center is near the somatosensory area of the tongue. I'm going to show you. This area of the cerebral cortex is responsible for the detection of somatosensory sensation from the tongue, and after this the pharynx area, and after this the taste center is there. I have not made that over here, but the taste center is near the somatosensory area of the tongue. The taste center, this is the taste center, and this is the somatosensory area of the tongue that is near. There is no connection between these two, but these are adjacent with each other.